Okay, let's go ahead and start. Welcome to the last week of classes. So just a couple of announcements, reminders, and such. There are 2 review sessions left. There's a review session tonight where we can cover both lecture material and lab material if people want to, as well as next Monday night, so the Monday in the first week of finals. And in case anyone has forgotten, hopefully no one has forgotten, the lab practical is this week. So on Tuesday and Thursday from at 9 and at 1 p.m. If it's possible, try to come to your own lab section. But if you have things that have come up and you need to go to different ones, that's fine. Just let us know. The exam will not take the full time. Nothing in microbiology lab ever takes the full 3 hours. So it should be fairly straightforward, but we can cover specific questions on it at the review session tonight. There is, as you're all aware, a lecture final exam the first Thursday of finals week. This is not what I erroneously put in the syllabus, so if you're going by the syllabus, please change your notes. It's actually going to be that first Thursday from 9 to 12 in here. This is also during that same 3-hour period that you can, if you wish, take one of the midterm retake exams. So it's a slightly shortened version of the first midterm that you've taken. It's not designed to be easier. It's just designed to be shorter. So there are less multiple choice questions that you can overthink and make mistakes on. So you just need to let me know which one, if any of them, you want to take. And the only reason for that is just so that I don't show up with only 10 versions of exam 2 and there are 12 of you that want to take it. So it just allows me to be prepared. So, and again, with the case with those midterm retakes, please email me just to let me know at the microbe mark email. I lose too many emails in my regular email account, so that's why I have this special one. Okay, so now let's move on and talk about the last section that we're going to talk about in micro this semester, and that's microbes in both health and in disease. So people probably know that we are greatly outnumbered in our bodies by microbes, but it's kind of astonishing to think that all of those microbes in there account for, on average, about 3 to 4 pounds of weight of each one of us. And just for reference, that's about equal to what the human brain weighs. And a lot of people have really begun to feel that the microbiota, the microbes that are associated mostly within our gut, but also within our skin and other mucosal membranes, but mostly within our gut, are really should be considered another human organ. Because probably one of the most exciting developments in microbiology in the last 10 years, and it's still very much in its infancy, are studies of the microbiota. And now what we realize is that the microbes in our gut have tremendous effects, not just on our digestive tract, but there are also some really cool studies that show that the human microbiota of the gut is associated with anxiety disorders, possibly even schizophrenia. Things like autism have been linked to specific disorders in the human microbiota. So it's a really interesting, fascinating, and still in its infancy type of science. So I can't really do it justice in the short time that we have left, but I just want to introduce it a little bit. Because for the most part, when we think about the human microbiota, we're talking about the gastrointestinal tract. So there's probably no place else in or on our bodies that has as rich and diverse a microbiota or an ecosystem as the gastrointestinal tract. You all know that certainly besides the more than 500 species of bacteria that are present in the intestinal tract, many of us have archaea, the other domain of life. And probably not too many of us left in this country, but not that long ago, all of us probably had multicellular organisms living in our intestinal tracts in the form of parasites. In the US and in Europe now, parasites are very unusual. But in many parts of the world, they're still a normal part of the microbial ecosystem. And it varies from different regions of the gastrointestinal tract, starting at the mouth, where you have mostly like, a huge collection of microbes, most of which die when they reach the stomach. The stomach is one of the probably most unappreciated roles that the stomach plays is in the protection of our body from infection. Because of the acidity in our stomach, most of the microbes that we swallow, which could potentially cause diarrheal diseases, which can be lethal in many cases, are actually killed by the acidity in the stomach. So it does a protective role for the lower portions of the gastrointestinal tract. But these portions of the tract, as you proceed from anterior to posterior, the numbers of bacteria and the species of bacteria richness actually increases tremendously until you get down into the colon, where you have this very anaerobic environment that has hundreds or thousands of species of bacteria, archaea. We won't go through the whole list of microbes. It's really kind of pointless. But some of the things that are partially unappreciated about the microbes there is that they actually serve a mutualistic role. We've already talked a little bit about how bacterial fermentation of fiber in the colon produces butyric acid, or butyrate, and that has a very important role in the metabolism of the cells that line the colon, and actually plays a role in protecting us from colon cancer. But microbes that are present inside the colon also produce a vitamin that we can't produce, vitamin B12, cobalamin. This is a vitamin that's a very important coenzyme in things like DNA synthesis. It's also very important in lipid synthesis. We can't make it. And if it's not in foods that we consume, it can be a problem. Also, vitamin K, a fat-soluble vitamin, we can't produce, but bacteria inside of our intestinal tracts are producing vitamin K for us. The contribution of bacteria today, in this part of the world anyway, to our diet of vitamin B12 or vitamin K are probably kind of small, because most foods that we eat are supplemented pretty heavily with vitamins. But probably in areas of the world today where still people don't eat highly processed, nutritionally enriched foods, probably a large portion of their vitamin B12 and vitamin K are coming from synthesis of those vitamins in the colon by this bacteria. So there's an important mutualism that goes on in there. And I want to talk a little bit about both health and disease at the same time, and how this colon microbiota or ecosystem actually plays into our own health. And so we'll start with a disease that's actually pretty rare, called, thankfully very rare, infant botulism. This is a problem obviously for infants, but it doesn't occur very often. There's only a handful of cases in the US every year, probably less than 20 cases in the US. But what this really shows you is why it's not a wives' tale that you don't feed children under the age of 1 year old, 1 year, honey. As it turns out, the bacterium that we all know that causes botulism, Clostridium botulinum, is actually a very widespread soil bacterium. It's not a human pathogen. We think of botulism as this terrible disease, and it is. But if you or I were ever to get botulism, it wouldn't be due to an infection. We wouldn't be infected by the bacterium Clostridium botulinum. Instead, what would happen is that some kind of poorly preserved, home-canned food contaminated with this soil bacterium would now reproduce inside that food and produce a toxin that we would consume. Our stomachs would kill the bacteria. We would never be colonized or infected by Clostridium botulinum. But that bacterium in that canned tomato or canned fruit produced a toxin which isn't destroyed by our stomach acid and is absorbed through our intestinal tract to inhibit acetylcholine release at neuromuscular junctions. So we get a terrible paralysis that can actually prevent us from breathing and dying. That's the normal way that you think of botulism. But in children, they can actually be infected. And it has to do with the fact that children are born, there's some debate about this now, but still, most people believe that children are born sterile. At the time that you emerge from the birth canal, you are a sterile vessel, and you begin to be colonized by microbes, obviously first from your mother, but then from your father and healthcare workers. And over the course of the first several years of your life, you assemble this very complex ecosystem in the intestinal tract. But for the first, certainly the first 6 months and possibly the first year, there's a very incomplete microbiota in the infant colon. So it starts off sterile. There are people who have shown some pretty interesting results where you can find microbes in the placenta. It's debatable whether those microbes were filtered out by the placenta or whether they were being used to selectively colonize the children. You can get into some real good debates at infectious disease meetings and microbiology meetings. But certainly, even if the child isn't born sterile, very close to that. So over the first couple of years, that microbiota develops. And it doesn't, a human doesn't develop what's generally considered to be the normal gut microbiota until 2 or 3 years of age. So what happens is that if Clostridium botulinum spores, so remember, this is a soil bacterium, so it has the ability to produce, as many soil bacteria do, the ability to produce these heat-resistant, chemically resistant spores that allow the bacteria to survive periods of low water or poor nutrition. If those, and they're frequently found in honey, bees bring them in and they accidentally and they get into honey. Now, when those spores get into the intestinal tract of a child, they can actually germinate and grow as vegetative bacteria because there's not very much competition. If the same thing happens in us, if we consume the same amount or more of honey, those spores may germinate inside of us, but we have hundreds or thousands of species of bacteria to outcompete the Clostridium. So it never grows to enough numbers to produce enough toxin to cause us any kinds of problems. The problem for children is that they don't have that type of competition yet. So this is why the toxin can now be absorbed into the nervous system of these children, because there's no check on the bacteria. So it has to do with a well-formed ecosystem. So prior to the development of that adult flora, these C. bot spores germinate, they grow as vegetative cells, and they can produce toxin. But as I said, this is a pretty rare event, but it does point out one of the major benefits to the microbial ecosystem that lives within our intestinal tract. It protects us, not just in this case, but in other cases as well. And so now let's go on and talk about a more severe, I shouldn't say more severe disease, this is also a lethal disease. I said it was more severe because I was thinking in terms of numbers of people infected. So there is a disease that you're all at least somewhat familiar with called pseudomembranous colitis. This is the disease that's caused by the anaerobic relative of Clostridium botulinum, called Clostridium difficile, or you just oftentimes hear it as C. diff. In an adult with a healthy colonic ecosystem, about 5% of people will be naturally infected by C. diff. And normally those numbers remain very, very low. What the actual role of C. diff is in a healthy microenvironment in the colon is unknown. What is known, however, is that people who are treated sometimes with antibiotics that kill off some of the competing microbes will allow the Clostridium difficile to overgrow. So some of their competing bacteria are removed by the antibiotics. So you might be treated for an antibiotic for a wound infection, for an ear infection or a throat infection. But if enough of the microbes in your intestinal tract are killed off, but C. diff is not, then some of the competition is removed and it can overgrow. And this toxin in large this bacterium in large numbers will produce a toxin, probably based on quorum sensing. The bacterium probably doesn't even bother to produce this toxin when it's at very low numbers, as it normally is. But when it overgrows, it now begins to produce this toxin, which is responsible for almost 30,000 deaths in the U.S. each year. That number varies depending on your source, but I think that's a fairly common or a fairly well accepted number. Now, this would be bad enough if we were just talking about people who were treated with antibiotics, on an outpatient basis and got these flare-ups of C. difficile. Many of these people would die. What's become a major problem is that C. diff has become what's known as a nosocomial or a hospital-acquired infection. So people in hospitals, as you know, are very sick. And there are large numbers of people in the hospitals that are being treated with antibiotics. So there's this large population of people in a concentrated area that have decreased ability to outcompete C. diff. And now you have a bacterium which has evolved to become hypervirulent. It's also a bacterium that has the ability to form spores. So unlike Clostridium botulinum, which is evolved to be a soil bacterium, C. difficile is actually highly evolved to be a member of the human gastrointestinal tract. It uses its spores to be able to spread from person to person. When it spreads from person to person, especially this new strain that's circulating in hospitals now, which is even more virulent than strains were 50 or so years ago, are hypervirulent because they have an increased ability to produce this toxin. It's a two-component toxin called TCDAB. But the other really evil thing about this is that this bacterium forms spores which can exist in the environment for 2 years, and they're very difficult to kill. So in hospitals, infection control is a major branch of hospitals. And one of the big things is hand washing, disinfecting areas. And so most of the things that disinfect what's referred to as the built surface in hospitals or people's hands are generally alcohol-based. And that does nothing to kill spores. So these spores are very difficult to eradicate. And so they're easily spread by healthcare workers from room to room. So it takes one person with a C. diff infection to potentially infect a large number of people in hospitals. So C. diff is a really major problem. But this toxin production inside the colon will cause severe damage to the colon lining. It actually does this by an enzymatic activity called glucosyltransferase. It inactivates small G proteins in the inside of the cell. If we have time, we'll talk more in another lecture about the mechanism of action of this toxin. But as I mentioned, the spores of C. diff have specifically evolved in association with humans. So that when a person in a hospital, for instance, ingests both, let's say they ingest both the vegetative cells of C. diff as well as the spores, the stomach does its job very well. The stomach will kill these vegetative cells, shown here in green, but the spore cells, or the spores, doing exactly what spores do, they're very resistant to chemicals. So they resist the acid destruction that happens to the vegetative cells, and they pass down into the small intestine. And you remember that one of the things that happens in the small intestine is the environment changes dramatically, because one of the things that we do to help saponify or solubilize fats, as well as to neutralize the acid coming in from the stomach, is to put in bile salts, bile acids from the gallbladder, made ultimately in the liver. The spores have actually used the bile salts as a signal that they've come home. What actually specifically causes C. diff spores to sporulate is the addition of bile salts. You can do this in the laboratory. The spores will sporulate, will grow into vegetative cells very poorly unless you add specific bile salts. These spores will now turn into vegetative cells. They've escaped the acidity of the stomach, so now they can live just fine until they find their way down into the colon, which is their natural home. So once in the colon, they can, if there's not enough competition from a well-formed microbiota, so if antibiotics have wiped out some of their competitors, the C. diff grows to very high levels and now has the ability to produce that AB toxin, shown here as two different proteins, one for attachment, or B for binding and A for attachment. Very similar in that way to the cholera toxin that we talked about earlier. But these toxins will actually get their way into these cells. By enzymatic activity, they will kill many of these epithelial cells, which results in a tremendous inflammation. So what's coming out of the blood vessel here are white blood cells, things like neutrophils, monocytes, cells that we call in times of inflammation, in times of infection, to help clean up the infection. But if they get out of hand, if the inflammation isn't cleared up, if the signals still persist, they actually will degrade and destroy some of the other cells, kind of a bystander impact. And they release this large amounts of fibrin and mucin, which forms what's known as a pseudomembrane. So this exudate, this pseudomembrane, will actually help to block the gastrointestinal tract. So this is from an endoscopy, and you can see these exudative plaques inside of a human colon. This is a severe case of pseudomembranous colitis. So now, rather than listen to me talk about this, I'm going to play a little bit of a video that's somewhat entertaining, but it's also very informative about how to treat Clostridium difficile. So one of the most clever ways that people have come up with, albeit a little bit perhaps disgusting, but hopefully this plays okay.
This ecology experiment is one of many that demonstrate how ecosystems with many species tend to be more resilient than those with few species. We're biased towards this particular grassland experiment because it's run by one of our writers, but strength in species numbers is also key to the health of ecosystems ranging from potato fields to rivers to the inside of your gut. In contrast, when a single species is dominant, it can cause problems. Take the case of Clostridium difficile, a natural part of your gut community that causes a severe inflammation of the colon when it becomes too abundant. This bacteria, often referred to as C. diff, was named difficile because it was difficult to grow in the lab, not because it's difficult to treat, but it is. C. diff infections often develop because these bacteria persist better and regenerate faster than other gut bacteria following antibiotic treatment for some unrelated health problem. Ironically, the standard treatment for C. diff is yet more antibiotics, which sometimes works, but in many cases the antibiotic resistance of C. diff means the playing field is simply clear of its competitors and its population can grow even more vigorously. So vigorously, in fact, that of the roughly 500,000 people infected each year in the US, about 1 in 30 die as a result, even after further drug treatments or surgery to remove parts of the colon. Out of desperation came the idea to entirely replenish the gut microbiome, not with probiotics, which only supply something like 1 living organism for every 10,000 bacterial cells already in the gut and just aren't powerful enough to make a difference, but rather with something more like the microbial equivalent of a blood transfusion. And just like a blood transfusion, don't try this at home. In a simple microbiota transplantation or beef transplant, someone with a healthy, diverse community of gut bacteria donates a sample to be administered to a patient with a C. diff infection. And in about 400 or 470 documented cases, the transplanted bacterial community quickly became dominant, causing the patient's digestive system to recover and stabilize, though scientists still don't know exactly how that happens. The human gut isn't the only ecosystem where this germ of an idea is taking hold, either. For example, transplanting soil from healthy fields to unhealthy ones helps knock back plant diseases and make soils more fertile, and transplanting water, snails, and duckweed from a pond to an aquarium helps maintain a well-balanced fish tank. At the moment, microbial transplant therapy for disease control resides at the fringes of medicine, agriculture, and home fish tank husbandry. The medicine is swing partly due to people being squeamish about their own and others' fecal matter, and insurance companies being squeamish about covering the cost of the procedure. But money also plays a broader role. New practices, even really good ones, have difficulty spreading without improved promotion. And that promotion often comes from places like the pharmaceutical, agrochemical, and home aquarium industries. Industries which promote only those things from which they can make a profit, like drugs, fertilizers, aquarium supplements, and other heavily engineered chemical cocktails. But who will promote the simple solutions? We will. From your C. diff to recruit, choose someone else's food.
I have to make sure the credits come because otherwise YouTube is apt to not let me post this. I keep meaning to check this one out. Do fetuses poop? I want to know the answer to that question, and I always forget to look that up. But actually, I mean, the FDA has not really approved the use of fecal transplants to treat C. diff, but they do accept the fact that it is highly effective, and now it's become very widely accepted. As a matter of fact, I was told recently by someone who I think has pretty good authority that all of the fecal matter used in prescription fecal transplants comes from students at MIT. I don't know why that is, but there are a group of students at MIT who are making lots of money donating their poop. Don't know why it can't be William and Mary. Okay, so now let's move from one portion of the body to another, and again, talking about both health and disease hand in hand, talk about how it is that the human microbiota protects us from another infectious disease. And so we'll start this off with kind of a question. So here in this graph is, on the y-axis, the number of reported cases of two different sexually transmitted diseases per 100,000 people, graphed over time, starting from about 1920. You can see syphilis, cases of syphilis caused by the bacterium Treponema pallidum, really spike around the time of World War II. So all of those boys going overseas, getting away, we're doing things other than fighting in the war. So syphilis becomes really high. Gonorrhea kind of parallels that, but was much lower. So here is the introduction of penicillin in the mid 1940s, and you can see that with that introduction, that syphilis goes down, gonorrhea goes down, but then something interesting happens here in the 1960s. Is the introduction of oral birth control. The pill is introduced. So what happens shortly after that is gonorrhea cases skyrocket. So think about that for a little bit. So here's the question to think about. We're going to get to the answer to this. This isn't going to be on the exam. Why is there a marked increase in the number of reported cases of gonorrhea with the development of oral contraceptives? Okay, so now let's talk about the normal microbiota in a different portion. So now we've moved from the intestinal tract. We're going to talk about the vaginal microbiota. So the normal vaginal microbiota in women is affected by hormones. So that ecosystem, the microbes that exist normally within the vagina, vary tremendously during the lifetime of a woman. Prior to puberty and then after menopause, the microbiota here is dominated by bacteria like Staphylococci, Streptococci, E. coli, and a group of bacteria called the diphtheroids. But at puberty and throughout the reproductive years, now with the advent of estrogen and all of the secondary sexual characteristic changes that accompany this increase in estrogen, one of the changes that occurs is that the cells that line the vagina, the vaginal mucosa, now those cells begin to produce an even larger amount of glycogen, that polymer of glucose. So that mucosa is a stratified squamous epithelium. It is designed to slough cells off the same way our skin sloughs cells off. And so as those cells, now rich in this glucose polymer, are sloughed off, they become a wonderful natural resource for microbes known collectively as the Doderlein's bacillus, named after a famous German gynecologist. You're a gynecologist. I can't imagine. But this was a bacterium that he studied, this Lactobacillus acidophilus, which is still a bacterium that's used today, becomes the dominant species in the microbiota during the reproductive years. One of the things that, and this is a gram stain of a vaginal smear, so you can see vaginal squamous cells that have come off, as well as these lactobacillus. So these are lactic acid bacteria. These are bacteria that can and do nothing more than glycolysis. They don't do respiration. So at the end of the glycolytic pathway, they produce massive amounts of pyruvic acid that they then ferment and turn into lactic acid. So this is what causes a very distinct change in the pH at puberty. So this lactic acid bacterium and its fermentation of glycogen results in a vaginal and cervical pH of about 4.4. So now, let's come back to the question of why it is that during the advent or post-advent of oral contraceptives, there was this big increase in cases of gonorrhea after oral contraceptives. Now, if I asked you this question before we talked about the estrogen story and the change in the vaginal ecosystem, you might have speculated that with the advent of birth control pills and the relief of the worry about undesired pregnancy, that possibly just having more sexual partners might cause this increase in gonorrhea. And that might be a reasonable hypothesis were it not for the fact that syphilis continued to decrease during that time. So if people were simply having more sex with more partners, then you would expect that sexually transmitted disease to have also increased. It really has to do with the changes to the hormones that go on during the use of oral contraceptives. Oral contraceptives, in fact, mimic pregnancy. And so what happens during that altered hormone balance is that the vaginal cells decrease the level of glycogen production. So the cells that are being sloughed off in the vagina now have less of this glucose polymer. And so the lactobacilli don't have as much natural carbohydrate source, so the pH rises. Without these bacteria producing large amounts of lactic acid, the local pH rises. As it turns out, acidity is a major downer for most pathogenic microbes, especially Neisseria gonorrhoeae, the bacterium that causes gonorrhea. It's actually really cool that nature has evolved to protect women from sexually transmitted diseases precisely at the time when they are most sexually active, during their reproductive years. So these pathogens, now in the presence of less glycogen and less lactic acid produced by these bacteria, not only will this bacterium that causes gonorrhea grow better, but also yeast, Candida albicans. So the increased yeast infection, which isn't charted on this particular graph, also traces its role to oral contraceptives as well. So the normal flora has evolved to protect humans during their most highly sexually active years, and it decreases at pre-puberty and postmenopausal, when hopefully in one case people aren't having sex, and in the other case when people are at least probably not having multiple partners in terms of sex. At least that's what I tell my wife. So now let's move on from that rather embarrassing portion of the lecture to talk about dental microbiology for those of you who are pre-dental students. So we're not going to go through the anatomy of teeth, but people obviously realize that caries or cavities develop because portions of the tooth can be dissolved by acid. So microbes play a major role not only in causing cavities and other oral diseases, they also play a major protective role in doing that as well. So bacterial biofilms, or plaque, as we know them, as we try to scrape them off of our teeth with dental floss and toothbrushes every day, those biofilms can ferment sugar into acidic compounds. So as we eat sugary snacks and don't adequately brush our teeth or floss afterwards, that plaque can now trap acids against our teeth, which can actually demineralize the teeth. So the plaque doesn't actually start off as pathogenic. It actually becomes pathogenic later on. Mouths actually have a healthy flora within them. So that immediately after brushing, or probably the better example would be coming back from your dental hygienist when you've been freshly scraped and polished and all of the microbes and plaque have been removed from your teeth, there's a flow of components coming out of the saliva which are going to form a pellicle, a protective series of proteins and glycoproteins coming out of the saliva that's going to coat the teeth. This is the first portion that, or the first step in the normal colonization of the tooth with bacteria that are either going to prevent dental caries or actually promote them. So these proteins will be bound to the dentin, the hydroxyapatite, within a few seconds of enamel contact. So here's that acquired pellicle. So these are proteins and glycoproteins that are coming out of our saliva that are attaching to the tooth surface. And then attaching to those will become the initial or the healthy microflora. So these are streptococcal species for the most part, Strep oralis, Mitis, Gordonae. These are microbes that now have adhesins or proteins that will fit, kind of like a lock and key, with various of these salivary proteins. This actually becomes a protective barrier for the tooth. Now, the early colonizers, the protective ones, are shown here, these streps and even some of the actinomyces. What happens later, especially in the case of poor oral hygiene and very high sugary diets, is that other bacterial species can become introduced. So Streptococcus mutans has always been thought of as probably the major cause of dental caries. That's actually becoming reinvestigated or re-questioned in the dental microbiology field. But we do know that Strep mutans is not present in normal, healthy plaque. It's only after people have consumed large amounts of sweets, haven't done good oral hygiene for many years, that this bacterium really becomes a predominant member of that biofilm. It's very cariogenic, meaning it produces cavities very well. It's part of a low affinity pellicle, meaning that it binds at low density to healthy plaque. So our healthy plaque is actually designed, has evolved to keep this bad actor out of the pellicle. And I've shown this slide before in an earlier part of the lecture, just to illustrate, again, like the gastrointestinal tract, the oral microbiota or microbial ecosystem is very complex, and its colonization occurs in waves. So you have these early colonizers, which only after these early colonizers shown here at the bottom, these streps, as they colonize, they provide receptors for other bacteria to attach. If those bacteria aren't removed by oral hygiene, then other bacteria will attach to them. And so eventually you build up a situation where you actually have some bad actors colonizing here, not only the strep mutans, but microbes that are associated with periodontitis or periodontal disease. The plaque itself, although it's healthy, can actually turn pathogenic. So if you look at, as in this graph, the pH of plaque over the course of a day, and you can see that here's where somebody ate breakfast, then they had lunch, then they had dinner, then they had coffee. If you measure the pH over time, you'll see that after eating, within that plaque, there is a drop in the pH. And remember, it's acidity, which is the enemy of teeth. It's what demineralizes and begins to break down the tooth material. But the plaque is very resilient, in this case of a healthy plaque. So you notice that the time spent below this critical pH, so below pH of about 5.5, tooth material can be dissolved in that acid condition. But you'll notice that in the case when the plaque is healthy, it rebounds quite quickly. And so brings this pH back up to above 6, where teeth are somewhat protected from acidity. Person eats again, it bounces back. This actually shows what happens when, as many of us do now, we frequently snack. So here's somebody, their plaque pH is being measured, breakfast, they had some kind of a sweet treat here, another one here before lunch, here's some more sweet treats, biscuits. So this, I think, was a British study, you can kind of tell from the afternoon tea and biscuit. But the point is that now that plaque, even though it's still maintaining at this point its capability to rebound in the pH, during the course of that day, that tooth is now exposed to a lower than pH 5.5 environment for a longer period of time. So demineralization can occur more frequently. This is part of the reason why you're not supposed to eat sugary snacks, and if you do, you're supposed to brush your teeth afterwards. So this healthy biofilm that's shown here in blue is always good, red is always bad, right? So here in the, actually that sounded more like a political statement, and I didn't mean it. But I think maybe the person who oriented this was probably Democrat and not Republican, right? But here you have this healthy biofilm, mainly of these commensal species, which actually kind of repels things like Streptococcus mutans. and some of the other microbes. So then the persistent low pH that occurs when people don't take care of their teeth and they eat lots of sugary snacks, that low pH kills many of those commensal bacteria. This is very much similar to the Neisseria gonorrhea vaginal microbiota story. pH at low pH tends to kill many bacteria. But it doesn't kill them all, because what it does is that it allows for other species that can tolerate acid. These are not part of the normal ecosystem, or at least they're not at high numbers in the normal ecosystem. These acid-tolerant bacteria, shown here in orange, begin to multiply, and they actually can produce more acid because they're tolerant of it, and so they produce a more persistently depressed pH, until eventually you have a very acidic biofilm. So this would be the strep mutans biofilm, a whole new biofilm. The healthy biofilm is gone. It's no longer capable of actively resisting something like strep mutans. And so now strep mutans, which normally doesn't compete very well against that healthy microbiota, doesn't have that healthy microbiota to compete with any longer. And so now this very new ecosystem forms, which has the ability to degrade teeth. So here's just a couple of slides. This is actually for next year. I just updated them. But one of the things that people realize now about dental microbiology and dental caries is that unlike other infectious diseases, where typically, if you have measles, it's caused by a virus. If you have strep throat, it's caused by a bacterium. These kinds of diseases like periodontitis and dental caries are now what's referred to as polymicrobial diseases, or dysbioses. So you have a disturbance in the normal ecosystem of the microbes. So what people have done is they've actually looked at different types of cavities. This is what's referred to as a white spot cavity, as opposed to a hidden dentin cavity. I'm not a dentist, I don't pretend to be one, but the different types of dental caries that occur actually have a different ecosystem within each one of them. So people have been able to look at the living microbes in the white spot type of carie as opposed to one in the hidden dentin, and they can consistently show a different microbiology associated with each one of those lesions. Now, it still remains to be determined whether something in this collection of microbes actually caused this specific lesion, or is it the ecosystem? Is it this lesion which allows this assemblage of microbes to survive in there? So there's not, right now it's just correlation, it's not causative. The same thing can be said here. Is there something about the environment of this hidden dentin cavity which permits these microbes to live and prevents the other ones? Or are these microbes actually causing that specific kind of cavity? But as you look at different types of cavities and larger numbers of them, so this is another white spot type of lesion, and you look at the different microbes, you can see that there's a fair amount of consistency, but still some variability in the microbes that exist in there, as opposed to a dentin damage. So here's a large crater on the top of this molar, as opposed to a hidden dentin one. So the microbial consistency between these differs. But you can see how complex that ecosystem is in each one of those. So figuring out who's doing what to whom is really difficult. This is I have great admiration for people who endeavor to do dental microbiology, because it's just too damn complicated. Okay, how much time have we got left? So what we want to do now is to move on and not so much talk about health, but to talk about disease. That's what's really interesting, right? So what we're going to do in the last part of this class is to talk about how bacterial pathogens cause damage to our cells. And we're going to talk specifically about how bacteria do this by the production of exotoxins. So we talked about endotoxins before. And remember, endotoxin is synonymous with lipopolysaccharide. Those two terms are used interchangeably. Exotoxins are actually secreted toxins. These are proteins that are released from the bacterium and have their effect on host cells without the host cell necessarily needing to be there. Now, most infectious disease lesions and symptoms are actually very complex. So most things that, fevers and all of the symptoms that are caused by most diseases are a combination of toxins, endotoxins, host inflammatory response. They're very complex. There are, however, a few diseases that are largely mediated by a single bacterial exotoxin. So these become, although they're difficult diseases to deal with, they're simple in the fact that the etiology of the disease is oftentimes a single secreted toxin. We'll talk a little bit about the diphtheria toxin. So diphtheria is a disease that hopefully none of you will ever see in your medical practices, those of you going on to medicine, but it's caused by bacterium Clostridium diphtheriae. Cholera is caused, that entire disease, caused by that single toxin. We'll hopefully get to that. Anthrax, botulism, tetanus. These are diseases that are caused by a single toxin. But these are kind of unusual. You know, unfortunately, most infectious diseases are not quite this complex, or are not quite this simple. So these bacterial exotoxins are, keyword here is secreted. They're usually proteins, but not always. And they alter the normal physiology or the integrity of the host cells. They're often used for vaccines. So probably most of you in here, hopefully all of you, have been vaccinated with what's referred to as the DPT vaccine. Diphtheria, pertussis, and tetanus is what DPT stands for. Now, oftentimes these toxins that are so bad for host cell physiology can be chemically treated to make them inactive. So you can treat them with a chemical like formalin, which will denature the protein slightly, so that, for instance, the tetanus toxin that causes tetanus, or the toxin that causes, I don't have it listed up here, the toxin that helps to cause whooping cough or pertussis. That toxin can be denatured slightly so that it doesn't cause the disease, it doesn't damage the cells, yet it's still recognizable by our immune system so that we can make an immune response against it. So these are what are referred to as toxoids, so inactivated toxins. So they're very useful in terms of vaccines. Now, in general, broad terms, when we talk about bacterial exotoxins or toxins in general, we can classify them based on their actions or their activities with host cells. So many protein toxins inhibit protein synthesis, host protein synthesis. We'll talk about some specific examples of that. Others will actually do membrane damage. You've seen evidence of this in the lab. So as you've seen hemolysis on the sheep red blood cell plates that we use sometimes in the laboratory, what's happening there is that those bacteria are secreting into their environment a toxin which forms a pore in a lipid bilayer of a host cell. In this particular case, it's the red blood cell, so the red blood cell lyses, and you can see that process of hemolysis. Just as a warning, keep that in mind for the lab practical exam this week. But these membrane-damaging toxins are released to kill things like white blood cells, not red blood cells, not what they're designed to. Other toxins will also activate second messenger pathways, and we've talked about this already with the cholera toxin. So inactivating the regulatory subunit of adenyl cyclase causes cyclic AMP levels to rise in cholera intoxication. So second messenger pathways are affected. They can affect the release of neurotransmitters. So this is the case of the Clostridium botulinum, Clostridium tetani. Those toxins actually affect neuromuscular junctions. And they can affect the host immune response, those super antigens. We're not going to get into these at all, but any of you who've had food poisoning where you've, well, unless it was salmonella, have probably felt the effects of this altered immune response and the super antigens. It's what causes that periodic retching or vomiting when you've eaten something contaminated with Bacillus cereus. These are super antigens that do that. And so when we come back on Wednesday, we'll continue on talking about bacterial exotoxins. I'll see you tonight at the review session.